Welcome to week 2 of lab. This week we're going to be looking at aseptic techniques. These are essential practices in the microbiology lab to help us prevent contamination and ensure that we're going to get some accurate results. Now, before we begin on the aseptic technique, let's just remember something from last week when we talked about microbes. Microbes are found everywhere. They thrive in environments where other life forms can't survive, and they exist in astonishing numbers. It's this ambiguous nature of microbes that make aseptic techniques so critical, because contamination will always be a risk because microbes are everywhere. So in lab, our goal is going to be to grow microbes, in particular bacteria. So when we talk about bacteria and we want them to grow, we're going to take them, we're going to place them onto a solid medium and get them to grow. But growth does not mean that they're going to increase in size, that each microbe cell is going to get larger. No. In microbiology, the term growth of bacteria means that they get larger in number, so that the amount of cells increases. When we grow microbes in lab, when we grow bacteria in lab and we put them on a solid media, when you first put them on there, you can't see them. They're invisible. So a term that you need to recognize is the term colony, because when we grow microbes in the lab and we get them to increase in numbers, eventually the number is going to become large enough that you can see it with the naked eye. And when it becomes visible to the naked eye, that's when we refer to it as a colony. Another term that you need to recognize is that when we try to grow microbes in lab, we want to have what we call pure cultures to study them. Why? Because then that means there's only one species, a single species of microbe on each of those cultures. And so that's what we want to maintain, is pure cultures in our microbiology lab. And therefore we need to know proper aseptic techniques in order to maintain those pure cultures. So if we're going to grow microbes in the lab, how are we going to do that? Well, microbes are living things just like everything else, so they're going to need nutrients to grow. So we're going to provide those bacteria with the nutrients through what we call a media or a growth media. Media's consistency can be either a liquid, semi-solid, or solid, depending on the amount of agar we use. And agar is the solidifying agent, and it's made from seaweed. So if it's a liquid broth, then there's not going to be any agar. If it's a semi-solid, it's going to have somewhere less than 1%. And if it is a solid agar, something like we use in a petri dish or a slant tube, etc., it's going to have more than 1% agar. So why then, if we're trying to grow in lab, is aseptic technique so important? One is because, as I already mentioned, it's an essential technique for microbiologists. But it is this aseptic technique which serves as fundamental principles for infection control in clinical lab settings, in patient rooms, in surgical settings, etc. So you need to understand the principles of aseptic techniques, not only for this class, but for your future in the healthcare field. So one of the reasons we need to know about aseptic techniques is because, as I said, we want to work with pure cultures. When it's not a pure culture and we get another microbe in our growth plate or whatever we're working with, then we say it's contaminated. Even if you know what that other species is, if it is unwanted, it is considered to be a contaminated sample. A lot of times we don't know what it is. So contamination can lead to inaccurate results or inaccurate identifications in a lab setting. Contamination can also occur not only in our sample, but of our work area or your person. So learning aseptic techniques will help prevent from passing pathogens off, and that will then lessen the potential for a health risk. So using those aseptic techniques in lab then will help you to learn how to remove or kill microbes from your person, like on your hands or the objects, so that when you're leaving that lab setting, you're not taking these unwanted things with you. So when we talk about aseptic techniques, the labs that you're going to be doing, even in simulation, they're going to be talking about inoculation loops, Bunsen burners, different kinds of medias. So the inoculation loops, those can be metal, which is the ones we're usually going to be referring to, because those are the ones we get to use over and over. But in some cases, they're plastic because they're a single use, one time only. The Bunsen burners, there's two different ways to incinerate or to heat objects like your inoculating loop. One is with an open flame from the Bunsen burner, but then there's also this thing referred to as an incinerator, which is like a small kiln. There is a ceramic tube on the inside, but it will get hot enough to incinerate any contaminants as well on your equipment. And then, of course, as I said, we're going to have the media that we're using in the labs. So it can be a petri dish where it's going to be a solid media, or it might be in test tubes where it's going to be a liquid media or a broth. So, how do we maintain aseptic techniques when we're working in the labs? So before we talk about transfer, we're just going to talk about a couple of steps first. So aseptic techniques using that inoculating loop. So again, we're talking about a metal inoculating loop that we would use multiple times. What we do to maintain sterility of a inoculating loop is we're going to flame it. In other words, we're going to put it up against really hot heat in order to incinerate any contaminants that might be on that loop. So if it's in the picture A here with the Bunsen burner, we're going to just stick it right into the flame, and we're going to hold it there until that loop, that metal tip, becomes red hot, and we're going to maintain it as that red hot for several seconds. Or we can stick it into the incinerator, which again, as I mentioned, was kind of like a kiln. It too will get it so hot that the loop will become red, and we're going to want to leave it in there for several seconds, again, to incinerate any contaminants. So once you have it doing that red hot glue for a few seconds, then you pull it out of the flame or you pull it out of the incinerator, and you're going to let it cool for a few seconds. And you have to let it cool, because if it's so hot to kill any contaminants, it's also going to be too hot and it'll kill your sample that you're trying to transfer or move or work with in lab. So you're going to allow it to cool for just a few seconds. And then how do you test that? Well, if this was in a lab, you would touch it up against the auger on the side of the plate if it was a solid plate, or if it was in the test tube, you would barely stick it into that liquid, and if it was sizzling, then you know it was a little too hot. Allow it to cool for just another second or two before you start your process and gather the sample that you need for transfer. The other thing that you have to be careful of when you're dealing with aseptic techniques and you're transferring, many times if you were in the wet lab, you would be starting with a liquid broth in a test tube. So when you're talking about maintaining aseptic techniques so that the liquid in here can be not contaminated, so it maintains that pure culture, you will run the test tube, the open test tube, over the flame a few times like this in order to heat up the air in there and the air around it to kill any contaminants. Because think about it, if you're going to be inserting a test, you're going to be inserting a loop into this test tube and going in and out. That's going to mix the air, mix the contaminants, and then that can cause contamination. So we're going to do this flaming. So look for that in some of the procedures. We'll talk about it a little here, but you'll also see that in your simulation labs. Another term just before we begin is the term inoculation. So in lab, many times what you're trying to do is move a microbe sample from one test tube or plate onto another plate or into another test tube. That process of moving that microbe and introducing it into a new environment is called inoculation. If you are moving that microbe, the term we use is inoculum to represent the sample or the microbe or the bacteria, in other words, that you are moving from point A to point B. And then in order to keep that media from not being contaminated, to ensure that your sample remains a pure sample, we're going to have to look at these transfer processes and try to maintain the best aseptic technique as we can. So when we talk about inoculation or transfer, you're going to often be going from a broth to a broth, or a broth to a plate, or a plate to a plate. So let's talk about using a broth and then getting a sterile, or getting an aseptic sample out for transfer. So notice we're going to start with an inoculating loop. We're going to heat that inoculating loop to get it sterile to begin with. So we're going to incinerate any contaminants might be on it. We're going to let it cool for just a minute, and then we're going to go into this sample broth, okay, that we have, that's going to have our single pure colony in there growing. So in order not to contaminate it, we have flamed the loop. Now we are flaming the test tube as we talked about before we insert the loop into the broth. Then we're going to pull that loop back out, and we're going to then stick it onto our plate, or this might be another test tube, and we're going to transfer that bacterial sample onto this by now just spreading that loop back and forth across this plate. A couple of things to point out. Notice that here, when we're working with the petri dish, we did not just take the lid off and set it on the countertop, because the countertops are contaminated. So then that would get our plate contaminated. Not only that, think about the air around it. So if things are falling constantly and from our air, if you leave that lid of that petri dish at this slant, then what will happen is any air particles, they'll hit the top of that lid and will not get onto the sample here, and so it won't contaminate your sample. So after you have removed your sample in an aseptic technique and you've brought it over to your plate and you've transferred it to your plate, then you're going to close your lid. One of the things you'll do is once you have this lid, remember it's still got sample on it, so, oops, excuse me, instead of going forward, now we're going to go backwards. So now you're going to close your test tube, heat it before you close it, and then you're going to flame your loop, all to be able to say, hey, we did that in an aseptic technique. So these are the quick steps that you'll review in the lab as well. Now, once you've gotten and transferred the sample to that plate, then what you're going to do again is use parafilm. Parafilm again is like a heavy duty saran wrap. We're going to wrap it around the edge, and that just prevents that plate from coming open or getting other contaminants. Once you've got the plate, then you're going to label it. I would often suggest labeling it first, but make sure no matter what that you're labeling your plate. And again, if you're labeling a plate in a wet lab, you're going to put your initials, the date, the course section, and what you put on there. So what was that sample? Was it a swab from your mouth? Was it a bacterial sample that you were provided by the instructor? Now you're going to label everything. Now one thing I want to remind you is that when you label, you're going to label the bottom of that Petri dish, not the top. Let me go back up here for just a minute. So you're going to label the bottom because that's where the sample is. That's where the media is. The top here might get lost. You might accidentally drop it. Somebody might accidentally bump your plate. So therefore, in order not to have to redo things, you label the bottom and not the top. Put your parafilm around it, and then you're going to incubate it, and when you incubate it upside down so that the media is up and the lid is down. So that is everything we're talking about in the aseptic transfer techniques in this video. Make sure you watch the other video on the colony, morphology, etc. And these are just to try to help get you oriented before you start your simulations in the Connect.